inhibitor can come out of the enzyme. That means this enzyme can be, if inhibitor comes out of the enzyme is free and can catalyze reaction, right? Kab? When this inhibitor comes out of the enzyme. So that means the condition is reversible. If the inhibitor bonds with the enzyme and forms the complex, then there will be no reaction. Reaction is blocked, enzyme has been blocked. But if enzyme is free and can catalyze another reaction, if the inhibitor unbound. So condition is inhibitor, if it combines with the enzyme, there are certain conditions for that, and if it comes out of the enzyme and leaves the enzyme unbound, then the condition becomes reversible. So that means this type of enzyme inhibition where the inhibitor reversibly binds with the enzyme. So until and unless the inhibitor is bound to the enzyme cannot work. But if the enzyme is been freed and by somehow the inhibitor comes out of the enzyme, then the enzyme can catalyze its normal reaction. So this type of inhibition is called as reversible inhibition. But in irreversible inhibition, the condition is different. That means the enzyme combines with the inhibitor E plus I and forms enzyme inhibitor complex. And this complex is formed irreversibly. That means there will be no reaction and it cannot be reverted back. That means the enzyme will be lost forever. It cannot carry out with its normal functioning after that. This reversible reaction is of three types. The reversible inhibition is of three types. Competitive inhibition, non-competitive, and uncompetitively. In case of irreversible inhibition, enzyme inhibition, the example is poison gas. There are certain poison gases like methyl isocyanide, they irreversibly combine with the enzymes and blocks its activity. So let us see the details of these three types of enzyme inhibition: competitive inhibition, non-competitive inhibition, and uncompetitive inhibition. Three types. So in competitive inhibition, that is the first type. Competitive inhibition of enzyme. What happens? The inhibitor combines with the enzyme and it competes for the active site of the substrate. So for example, it is the enzyme and we have a substrate here. The substrate wants to go to the active site of the enzyme. The active site of the enzyme is this. This one is the active site of the enzyme. So for example, this is the active site of the enzyme. But there is an inhibitor. This inhibitor, say for example, this inhibitor, which structurally quite resembles the substrate, it is inhibitor. And it combines with the enzyme. It competes with the substrate. This inhibitor is competing with the substrate for the active site. And the number of inhibitor molecule is more than the substrate. So the inhibitor has got more chances to bind with the enzyme. So finally there will be enzyme inhibitor complex. So there will be enzyme plus inhibitor, the substrate is left out. So it leads into the formation of enzyme inhibitor complex. And then there will be no reaction. That means normal reaction which has been catalyzed by this enzyme will not take place. This is competitive inhibition. We call this type of inhibition as competitive because the inhibitor is competing with the substrate. It competed with the substrate for the active site of the enzyme. So that means this inhibitor have got structural similarity with the substrate. It is not accurate 100% similar, but quite similar to the substrate. Now in competitive inhibitor example, let us see one more example of competitive inhibition. There is an enzyme succinic dehydrogenase. Succinic dehydrogenase. So this is the enzyme. The normal substrate for it is succinic acid. So this succinic acid combines with the succinic dehydrogenase. So this is the substrate. But sometimes malonic acid is there. We call it as malonate. It is the inhibitor. So we have another inhibitor. So this is malonate. So malonate competitively inhibits the action of the enzyme succinic dehydrogenase. This is a broad example of it. Now, if we look ahead with the characteristics of this competitive inhibition, there are two important characteristics of this competitive inhibition. One of the characteristics is in competitive inhibition, so I am writing it in short form. In competitive inhibition, the Km value increases. Okay, the Km value increases, but Vmax remains constant. So this is the key manifestation or key points that has to be remembered for the competitive inhibitor. So if you see this graph, then we find a graph like this where velocity is increasing this way and substrate concentration is increasing on x-axis, y-axis. The rate of enzyme action is versus substrate concentration. So where the bending is occurring, if we go that point where the bending occurs, that becomes V max. So half V max, at half V max, if we are supposed to locate the substrate concentration, so that substrate concentration, that point where the perpendicular meets on the x-axis, is the Km value. So if this green color is showing the Km value without inhibitor, all right, so with inhibitor this value will increase. So let us see how. This is the case with inhibitor, red one. So blue one without inhibitor and red one with inhibitor. So this is with inhibitor, the plot with inhibitor, and it is without inhibitor. So here we see that the Km value without inhibitor, with inhibitor, it is like this. I will draw a perpendicular from here with red one, then the Km value will lie somewhere here. That means there is shift in the Km value, so it increases. So Km value increases. This Km increases here because of the difference in the steepness of the curve. So the red one is with inhibitor, and the blue one is without inhibitor. So what do we notice here? This curve show a different variation without inhibitor and with inhibitor. So what do we notice here? The Vmax value and the half Vmax in between these two, the Vmax value remains the same for both. That is, the bending is occurring at the same place. But because of the difference in the steepness of the curve, the half Vmax increases. That means this half Vmax will lie in different places. So the Km value, increases. So Vmax and half Vmax, they both remain the same. But this Km is increased from this side to this side. Alright, so this is the case with graph with competitive inhibitor. So this inhibitor will be actually a competitive inhibitor. This will be inhibitor. More correctly, we call it as competitive inhibitor. And that's why we say with competitive inhibitor, the Km value increases and the Vmax remains constant. Also, half Vmax remains constant. These are the two characteristics that has to be learned for the competitive inhibitor. So this inhibitor is substrate analog. We also call it as substrate analog. That means the inhibitor looks like the substrate. Alright. So quickly, I am going for a revision of the competitive inhibition. The competitive inhibition is a type of inhibition where the inhibitor quite resembles the substrate. So it is a substrate analog for the enzyme inhibition. So this inhibitor, if present in higher quantity, competes with the substrate and forms enzyme inhibitor complex, and then there is no reaction. But it is reversible. That means when the inhibitor will be taken out, so somehow this enzyme will become normal. It can carry out its normal activity. One of the example is succinic enzyme, that is the enzyme, it is being inhibited by malonate, which is a competitive inhibitor for succinic acid. Now, in case of competitive inhibition, the Km value increases. That means we are finding a shift from this side to that side on the substrate line, but the Vmax and the half Vmax, they both remains constant. Okay, now this is the basic information and idea about the competitive inhibition. But here we are more concerned with the clinical manifestation of this. So let us see how this competitive inhibitors can be used by us as anti-metabolite in chemotherapy. That means uses of competitive inhibitors in medicine. Competitive inhibitors in medicine. That means how in medical science we can use the competitive inhibitors for different issues. Example 1. There are certain anti-metabolite or chemotherapeutic drugs which are used as competitive inhibitors. Example 1. First, we have p-benzoic acid. P-benzoic acid. Usually we call it as PABA, PABA. Now this p-benzoic acid is converted into folic acid inside the bacteria. It's converted into folic acid. It is a normal in case of bacteria. Now what happens, this folic acid, it is responsible for growth and development of bacteria. What is it? What is the function? It is responsible for normal growth and development for bacteria. The enzyme which converts, there is an enzyme which converts PABA into folic acid. Now, there is a substrate, there is a competitive inhibitor. This is sulfonamide. This sulfonamide blocks this enzyme. It blocks the enzyme responsible for the conversion of p-benzoic acid, that is PABA, into folic acid. If this enzyme is blocked, then what will happen? If this enzyme is being blocked, then what will happen? If it is blocked, then no folic acid formation will be there in the bacteria. There will be no folic acid formation in bacteria. So bacteria will die. So that's why this is used as to destroy the used in infections, to destroy the bacteria. It's a competitive inhibitor used to destroy, like used to kill simple bacteria. So help in infection control. So this sulfonamide is the first drug which is used as a competitive inhibitor. It is used as competitive inhibitor to control the infections. So there are questions which comes in your medical also, that discuss any 4 drugs that act as competitive inhibitors, and or discuss any 4 or 5 drugs which are used as competitive inhibitors as antimetabolite in chemotherapy. So this is the first drug, sulfonamide, which is used by us to control the infections. And what does the sulfonamide do? The sulfonamide blocks the enzyme which is responsible for the conversion of PABA into folic acid inside the bacteria. This folic acid is responsible for normal growth and development. So if we block this enzyme, and it can be achieved by sulfonamide, so what will happen? The bacteria will be killed because folic acid will not be formed there, and that's why we can control the infection. So if a person is having some bacterial infection, then this sulfonamide are usually used as a infection controlling drug. So this is first example, and this sulfonamide is a competitive inhibitor. All right. Now second example, let us come on to the second example. When we write second example, it is aminopterin. So this is the second drug, aminopterin. Example of aminopterin is methoxate. Methoxate. Now what does this do? It is used for controlling cancer. It is used to control cancer. Then these amikopterins or this methoxate is a competitive inhibitor. Actually, it is competitive inhibitor. I am writing it short, competitive inhibitor. Now what happens? In our body, the dihydrofolate, it is a substrate. Dihydrofolate, it is a substrate. It is converted into tetrahydrofolate. and this is converted into this tetrahydrofolate is required for the formation of nucleic acid, and the nucleic acid cause cancer somehow. It is responsible for nucleic acid formation, that is DNA. Alright, so what we notice here, there is an enzyme dihydrofolate reductase which do the first reaction. Dihydrofolate reductase. So this is the enzyme which carry out the reaction, that is conversion of difolate into tetrafolate. This tetrafolate is required for it is responsible for the nucleic acid formation, and this nucleic acid which is formed in extra quantity is responsible for cancer. It causes cancer. If you have studied about the cancer, then it is uncontrolled protein synthesis. And who is responsible for protein synthesis? The DNA. If you have got excessive or uncontrolled DNA formation, that may lead into the uncontrolled protein formation, and that leads into the cancer. So this reaction is responsible for cancer somehow. Now how do we control cancer? How do we control this cancer? By this methoxate, we can do that. So these, I am writing the competitive inhibitor. These aminopterin actually block this enzyme. This block the dihydrofolate reductase, and when this enzyme is been blocked, there will be no formation of tetrahydrofolase. So there is no formation of non-formation of FH4, that is tetrahydrofolate or tetrahydrofolate. So if the first enzyme has been blocked, if this enzyme has been blocked, the reaction will be blocked. There will be no formation of tetrahydrofolate, and then there will be no nucleic acid formation, and then we have got no cancer. Cancer can be prevented somehow. Or we can go for one correction over. It is not ido, it is hydrofolate, tetrahydrofolate. Tetrahydrofolate. All right. So the use of aminopterins, or example is methoxate, it is used for controlling cancer somehow. This can be one achievement. So these aminopterins are used as a chemotherapeutic drug to control the cancer. So we have done two drugs now until now. First one was sulfonamide, which is a competitive inhibitor, and this aminopterins, which is again a competitive inhibitor for dihydrofolate. So this is the substrate, and this is competitive inhibitor. So these two are competing for the enzyme dihydrofolate. Now let us take one more example, third example, example 3. We also use another drug, that is allopurinol, which is used in the treatment of cancer. So let me write the name of the drug first. The drug is allopurinol. It is competitive inhibitor. Now what does it do? It is used in controlling gout. It is used in the treatment of GOT. And how does we do it? In our body, we have got a normal reaction that in which hypoxanthine is converted into uric acid. This hypoxanthine, which is a substrate, is converted into uric acid in our body. And this uric acid, when accumulates in high concentration, it causes GOT. That means this uric acid will accumulate in the joints and will cause stiffness of the joints, and that is called as GOT, one of the major reason for this GOT. And this reaction has been catalyzed by xanthium oxidase, or we call it as xanthino oxidase. This is the enzyme. Now, the competitive inhibitor, that is allopurinol, this is competitive inhibitor for this hypoxanthine, go and blocks this enzyme. It go and blocks the enzyme. If this enzyme is blocked, there will be no this. If this enzyme is blocked, then there will be no uric acid formation. Then there will be no uric acid formation or synthesis. Now, if there is no uric acid synthesis or formation, then we can prevent the GOT. The GOT can be prevented. Alright, so this is example 3, allopurinol, which is used as in the treatment of GOT. It is a competitive inhibitor. Now come on to another example, example 4. We are doing competitive inhibitor and its clinical manifestation. Example 4. There is one more compound which is used as a competitive inhibitor for the control of cholesterol, and this is levastatin. So levastatin is a competitive inhibitor. It is a competitive inhibitor and used in the treatment of cholesterol formation. Used in treatment of, we call this condition as atherosclerosis. Atherosclerosis is accumulation of cholesterol in the body around blood vessels. Accumulation of cholesterol. So if the cholesterol will accumulate in the blood vessels, then what will happen? It can lead into the high BP and also the further troubles like brain stroke and all that. So in our body there is a reaction in which there is a precursor substance. This is the substrate. I am not writing the exact name of that precursor. It is converted into cholesterol. When we will be studying about cholesterol, then we will do that. So cholesterol formation is there in the body with the help of HMG coenzyme A. This is the enzyme for the formation of certain precursor substance into cholesterol. But we have this levastatin. Now, what does the levastatin do? This levastatin act as a competitive inhibitor, and it competes with this precursor substance and blocks this enzyme. This blocks this enzyme. So if the enzyme has been blocked by the levostatin, there will be no further formation of, what will be the result? There will be, if this enzyme is blocked, then no further formation of cholesterol. There will be no further cholesterol formation. So like this we can control atherosclerosis by preventing the formation of cholesterol by this levostatin. So levostatin actually is acting as competitive inhibitor, and it is helping us in the control of atherosclerosis. Now let us one last example we have to see here, which is used as a competitive inhibitor, and this is in the treatment of hypertension. So example 5. There are the 5 drugs which are used as a competitive inhibitors, and they help us to combat certain medical manifestations or medical diseases. Say for example, we have got captopril. It is another substance which is used as a competitive inhibitor. Along with it, enalapril, lisinopril, they are all competitive inhibitors used in the treatment of hypertension by lowering the BP. They are all used in treatment of hypertension, by lowering the blood pressure. If we are having blood pressure, then it can be lowered by these competitive inhibitors. Now what does they do? They all blocks an enzyme which is responsible for converting angiotension into tensinogen into angiotension. So these competitive inhibitors blocks certain enzyme which are used in synthesis of angiotensin. And in physiology we'll study about this angiotensin. It regulates blood pressure, or actually it increases blood pressure. It regulates and somehow increase also blood pressure. So during hypertension the BP is increased. So if you block the synthesis of angiotensin by blocking the enzymes used in the formation of angiotensin, then the BP can be pulled down, and we can get rid of the hypertension. So these are the examples of competitive inhibition which are used in medical science to combat certain disorders or certain diseases or abnormalities of the body. So let me quickly revise this competitive inhibition. The competitive inhibition is a type of reversible inhibition where the inhibitor binds irreversibly. Alright. In competitive inhibition the Km values increases but the Vmax remains constant. And these are the certain drugs which are used as a competitive inhibitor to combat certain medical problems. Sulfonamide, which controls the infection by blocking the enzyme for the formation of folic acid. Aminopterins, which are used as competitive inhibitors, which blocks the dihydrofolate and blocks the formation of tetrahydrofolate, and that's why the cancer can be prevented, because the tetrahydrofolate is used in the synthesis of nucleic acid, especially DNA, and then excessive DNA formation leads into cancer. Allopyrinols, it is used in treatment of gout, where hypoxanthine is converted into uric acid. If you want to prevent the formation of uric acid, we have to block this enzyme, hypoxanthino oxidase. So allopurinol, it is competitive inhibitor. This allopurinol themselves convert into hypoxanthine also, and that's why this type of inhibition is also called as suicide inhibition. It is also called as suicide inhibition, because this allopurinol also helps in the synthesis of hypoxanthine. Now, another example is levostatins, which are used in the treatment of atherosclerosis, that is the accumulation of cholesterol, especially in the blood vessels. The enzyme responsible for this formation of cholesterol has been blocked by levostatins. So, levostatin is another example. Then coprolprils, enalaprils, and cineloprils, they are the competitive inhibitors used in the lowering BP by and then they are useful in controlling hypertension. They block the enzymes which are used in the formation of angiotensin. So if it is not formed, then the blood pressure can be lowered. Now this is all about competitive inhibition. Now the next is